Hey guys, Kenna here, and today we're going to be talking about shared biochemistry. So, this is going to kind of tie together what we did last term and really kind of focus on bringing together the pieces so that we can go ahead and talk about genetics and evolution in this semester. So, let's begin with our essential questions. Number one, what shapes the characteristics of all living things? And you may think you have the answer to these already, and that's great. But we want to make sure that we're all talking about the same things and we understand some of the same concepts. Number two, why can living things decode the information in each other's genes? Maybe you didn't think about this before. Maybe this is something new, or maybe you already knew this. Let's talk a little bit about what about the genetic code allows this to happen. Number three, how do DNA and proteins relate to each other and to the functions of the cell/organism? This is a key piece that we wanted you to take away from last term. And if we didn't, we're hopefully going to bring up some key pieces here. And if we did, awesome. It's great to go ahead and review these things. One thing that I noticed when we were going ahead and talking about transcription, translation, and protein synthesis is that there were a few kids who started really kind of thinking that DNA somehow transformed into mRNA and mRNA somehow transformed into tRNA. That's not what happens. What happens is we do this complementary base pairing where one chain is created as a copy of the other. So the bases match, but we're not converting one thing into the other. When we talk about it in terms of doing transcription, we're converting the code from DNA into mRNA, which changes T's to U's, but we're not actually changing the DNA. We're making a copy. That copy is complementary to the original DNA sequence. When we match this up with tRNA, same sort of thing. We're not changing anything. The mRNA matches up with the tRNA. So then we get a triplet codon that's matching up with a triplet anticodon, and that's going to give us our particular amino acid. Okay, so let's go ahead and watch a little video and kind of get some of these pieces hopefully in place.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
So as we start taking a look at this idea of reading genes, remember most of the heavy lifting in your body is done by proteins. Proteins do most of the work, but all the instructions are coded for inside the DNA. So we have to use that DNA to build the proteins to do the actual job. And so this relationship between DNA to proteins is a really important one for both genetics and for evolution. Now, you've heard a lot about the term genes, I'm sure, and no, we're not talking about the ones you wear. We're talking about the ones in your body that make you who you are, at least to some extent. So, what are DNA versus genes? How are they the same and how are they different? Now, DNA, hopefully you remember, is all of the genetic information that you have. Okay? It's all coded for in DNA. And we call this your genome. But what we do when we do transcription and translation is we go ahead and take a small section of that genome, and that's what we call a gene that we're going to go ahead and copy and make mRNA and eventually a protein from. So, a gene is a sequence of nucleotides in DNA that codes for a particular protein. So, let's watch this next video, make sure we understand these concepts.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order. But small differences in the sequence of the bases in our genes make each person unique.
Okay, so a couple quick things here. So it really focuses on DNA, but know that we've talked about RNA as well, and there are some similarities here. Remember that DNA is double-stranded, so there's 2 chains, and it's twisted up in that double helix. Those strands, remember, run opposite directions, so one strand is flipped from the other strand. When we copy this, we're only copying off one strand, the template strand, to make our mRNA transcript. When we do this, remember mRNA is only single-stranded and must be edited, keeping the exons expressed and removing the introns to make the final mRNA transcript that is sent out to the ribosome to make your protein. So the human genome is all of the genes. Okay, they're in the same position on the same chromosomes, but your version of a gene, and we'll talk more about this a little later when we get to genetics, your version of the gene is what is unique to you. Okay, now there may be some small mutations that you have that nobody else in the entire world has, and that makes yours completely unique. Or you may have a copy that matches exactly to one of your parents. We'll talk more about this again when we get to genetics in a little bit. But all DNA, whether it's yours, whether it's a tree, whether it's a bacteria, is the same stuff. If you take a look at the picture here on the right, this is a test tube that actually has DNA in it, and you may have done a DNA extraction, maybe from strawberries or bananas at some point in your career. But you can't tell what organism this DNA in the test tube came from just by looking at it. And that's because it's the same for every single organism on Earth. That little kind of ball of snot, right, is the DNA for that organism. And since we said, in terms of the characteristics for life, that all living things have DNA, whether it comes from you, a pea plant, your pet rat, it's all the same molecule. It's the order of those particular letters or bases that we've discussed in the code that makes each organism different. How many, what sequence, what particular gene are they found on? In the human genome, the order of the building blocks in a strand makes up a sequence, and we can read that DNA sequence like letters in a book. In fact, we know the sequence of the entire human genome, all 3 billion letters. That's enough information to fill roughly 1,200-page books. Contained within the 3 billion letters of the human genome are about 21,000 genes. Most of our known genes code for proteins, but some code for RNA molecules. Remember, we have to create our mRNA, tRNA, and rRNA. Okay. Now, when we start looking at these still have to be manufactured. So it's pretty cool when you stop to think about this because while all humans have the same genes arranged in the same order, and this should really kind of put things into perspective. You have far more in common with the person sitting next to you, the person you're talking to on the phone, or the other people in this class than you do different. More than 99.9% of our DNA sequence is exactly the same. But the few differences between us, all 1.4 million of them, are enough to make each one of us unique. On average, a human gene will have 1 to 3 bases that differ from person to person, and these differences can change the shape and function of a protein, or they can change how much of a particular protein is made, when it's made, or where it's made. Okay? So these tiny changes can have a significant impact. And even when we start looking at twins who begin as a single cell, so identical twins, by the time they become adults, they've accumulated enough mutations or changes in their DNA that they too are unique from each other. Now, that structure that this is stored in is really important, and that's the double helix. And most of you picture the double helix, not the snot we saw in the previous slide, but this spiral, beautiful spiral structure of the double helix. And we have not always known how special or how cool that particular structure was. I will go ahead and link to the video we watched last term over the history of the discovery of the double helix. So you might want to go ahead and take a look at that if you don't remember the structure or if you don't remember how powerful the discovery of that structure actually was. But today I want to go ahead and focus a little bit more on looking at proteins and what proteins exactly do. Okay, we've talked about this before when we were looking at macromolecules, but we need to refresh and think about the relationship of this to the DNA. So let's start by watching this video.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
So we spent a lot of time last term delving into the relationship between structure and function. And this is another excellent example of that. The video also identifies how cells are specialized. Not every cell makes every protein. They only make the proteins that it makes sense to have in that particular cell based on its job. This is a great example of both structure and function and specialization. So, as it said in the video, and I've been kind of telling you this all along, proteins are the big workhorses of our bodies. They do so much for us in everyday living. Almost every task your body performs, done by a protein. And proteins make up about 42% of the dry weight of our bodies. The protein collagen alone, which holds our skin, tendons, muscles, and bones together, makes out about a quarter of the body's total protein. That's a lot of protein. All of our cells and even blood are packed with protein molecules. The watercoloring you see on the right side here is by painter David S. Goodsell and shows part of a red blood cell at the bottom left, filled with hemoglobin molecules. The upper half of the painting shows blood serum and containing yellow, Y-shaped antibodies and other proteins. In purple, proteins poke through this blood cell's membrane surface. We started looking at the lipid bilayer and looking at the fluid mosaic model. Proteins just do so much for our us and our survival. But these proteins have their instructions embedded in the DNA. And so that DNA has instructions that are readable by every organism on the planet. And so we can take proteins sometimes from one organism and insert them into another, or take the DNA for a particular protein and insert it into a new organism that will now make that protein it didn't previously make before. Proteins are pretty amazing, versatile things. Different arrangements of the amino acids can make proteins that are really strong, as the silk fibers we mentioned at the beginning of today's lecture, or flexible and elastic, as the elastin in our skin. And like pieces of a jigsaw puzzle, proteins can interlock with other molecules. Remember, we talked about that quaternary structure and how we have these subunits working together to do the final job of the protein. Okay? Antibodies in our blood have a unique arrangement of amino acids at its tip that can attach to a very specific pathogen, marking it for destruction by the immune system. Okay. This is a tag and conquer system. It's pretty cool. So it's involved in your immune system, your physical structure. It's going to go ahead and carry oxygen through. The, the major functions of what you need to survive are primarily done by proteins. Now, proteins work in groups. They need to physically interact with each other and with other molecules to do their jobs. These interactions might activate an enzyme or turn on a gene or communicate a message from one cell to the other. Do you remember the internal external regulators, the cyclins we talked about with respect to cancer? Interactions between proteins depend not just on their shape, but also on their chemical properties. Positively, negatively charged amino acids are attracted to each other. Hydrophobic residues cluster together away from water. These physical properties allow proteins to interact in very specific ways to do unique jobs. Proteins also change shape. They don't always look the same way that they did when they were first created. Cells are constantly moving, they're full of motion, and this is largely driven by proteins. Many proteins are flexible and dynamic. Motor proteins, for example, will bend and swing to literally walk across the cell's cytoskeleton. Things like kinesin. If you've ever seen one of those animated videos showing you the inside of the cell, the little legs that are walking across the tightropes, these are kinesin. And when the neurotransmitter acetylcholine binds to a receptor, the entire protein molecule shifts, causing a hole to open up and allow passage of materials. Sodium ions to pass through the opening start chain reaction that'll fire nerve signals across the brain. This is all possible because of proteins. And proteins aren't a one use thing, they're constantly recycled. Like us, plants and animals are made up of proteins. When we eat them, we eat protein. High protein foods like beans, meat, fish, cheese, eggs, and nuts give us not only energy from the bonds that we break down, but they also give us the building blocks that actually make us grow, help us to maintain a healthy body by repairing our internal structures. So the proteins we eat are broken down into their tiny little amino acids. Remember those monomer building blocks we discussed? And we constantly reuse these to build new proteins. What kind of proteins? Oh man, the list is enormous, but here are some examples. So we can start out with, let's say, enzymes. Okay. Enzymes build and break down molecules. They are critical for growth, digestion, and many other processes within the cell. Without enzymes, chemical reactions would happen too slowly to sustain life. Remember, one of the key things we talked about was that enzymes are going to lower activation energy so chemical reactions can occur. Next, let's move on to structural proteins. Now, structural proteins strengthen cells, tissues, organs, and more. Nature can build some pretty amazingly strong materials. In fact, your body is primarily built outside of the water, right? It's built of these structural proteins, things like collagen, keratin. Okay. But when we start looking at these, we also talked about our cyclins, those regulatory proteins that help to signal between cells and within cells. Okay. These allow communication from one cell to the next. They signal, have receptors, and relay proteins work together to get information from outside of a cell to the inside. Not only do we have to do communication, but we have to regulate certain cell processes, turn genes on, things like that. So regulatory proteins bind DNA to turn genes on or off. We'll talk about methylation a little later on in this term. These are used to build the proteins that we need when we need them. This is turned on and off by other proteins. We already talked extensively about the next group. These are your transport proteins, the things that are embedded in the lipid bilayer. These help move molecules and nutrients around the body in and out of cells. So you see these in not only the cell membrane, but also in the nuclear membrane and many of the other membrane-bound organelles. Sensory proteins are there to help us learn about our environment. They're what allow you to detect light, sound, touch, smell, taste, pain, heat. Some organisms can even detect electricity or magnetism and have organs inside their body that are sensing these things using proteins. Motor proteins, don't want to oversell these. They're exactly what you think they are. They keep our cells moving, changing cell shape. We think about like myosin and actin. These are what are going to go ahead and allow our body to flex and contract. We have our defense proteins. These are the ones that are involved in helping us fight infection, to heal damaged tissue, even to evade predators. These responses occur internally. And then lastly, we also have some storage proteins. These help us to maintain nutrients and any rich molecules for later. And that's just a small spattering of the groupings or categories of different types of protein, not an extensive list by any means. So let's think about what we learned when we were going over protein synthesis. The DNA is connected to proteins through the copying of a gene. Remember, a gene is a sequence of nucleotide bases in DNA that codes for a particular protein. We begin with transcription that copies the DNA into mRNA. Genes containing instructions for assembling proteins get turned into amino acids. But that starts with the mRNA, which carries the copies of these instructions to the ribosome. The ribosome, the mRNA will be matched up with the transfer RNA or tRNA, and they join together to give you specific amino acids in a process known as translation. These eventually fold into a protein, and this whole process is known as protein synthesis. Now let's take a look at a particular example of how this works. The example I'm going to give you here today is that of fireflies. I don't know how many of you have actually seen fireflies, but it's pretty spectacular when we start thinking about all the cool things that they do. So let's start by watching the video.
How a firefly's tail makes light. First, cells in the firefly's tail make luciferase protein. Then, luciferase carries out a chemical reaction that releases light. The light comes from specialized cells in the firefly's tail. Each cell holds a copy of the firefly's entire genome. One of these genes is a set of instructions for building the protein luciferase. A molecular machine called RNA polymerase makes a messenger RNA copy of the luciferase gene. The gene can be copied many times. The mRNA attaches to a ribosome, the cell's protein-making machinery. The ribosome reads the molecular code of the mRNA to build a luciferase protein, one amino acid at a time. Multiple ribosomes can read each mRNA copy to build many luciferase proteins at a time. When the ribosomes finish reading the mRNA, the proteins are released, and they fold into their final shape. The ribosomes can read the mRNA again to make even more luciferase. Luciferase is an enzyme that catalyzes a chemical reaction between a substance called luciferin and oxygen. This reaction uses energy from ATP, and it releases light. Luciferase can catalyze this reaction again and again. The action of many luciferase enzymes acting at once makes a light bright enough to attract mates and warn off predators.
So, one of the main reasons we talk about is that fireflies glow to attract a mate. Males and females of the same species will flash signals back and forth as a way to communicate, each firefly species having its own particular pattern of flashes that they make. For example, the fireflies of one species will fly around in the night sky and dive steeply just as the flash begins and turn upward to make a distinctive J shaped pattern. Female fireflies hang out on tree branches or in the grass while males fly around showing off their best flashes. When the female recognizes the flash from a male of the same species, she answers with her best flash. Thus, they are introduced to each other. Another reason that fireflies glow is to avoid predators. Fireflies are filled with a nasty tasting chemical called, I'm going to butcher this, lucibuffergens. And after a predator gets a mouthful, it quickly learns to associate the firefly's glow with this bad taste. So not only does the flashing help to attract a mate, but it also warns predators to stay away. One species of firefly that can't make its own chemical acquires it by eating others that can. To lure victims, these fireflies mimic the flashing patterns of another species, and when the unsuspecting male approaches to find a mate, he instead becomes a snack for the tricky firefly. So let's watch. We got one more video going over this, and then we'll go ahead and move on from there.
So we're out at Goshen Ponds. We're out here today looking for fireflies. People don't think they're here in the West because fireflies are traditionally an eastern species. We found flashing fireflies in Utah. That's a pretty big deal, and can't wait to get them back to the lab and start to analyze them and see what species they are. The reason why people like fireflies so much is that they produce their own light, and it's something that captivates us. And beyond that, the light's beautiful. They come in greens and yellows and oranges. They come out in large numbers, and it's a pyrotechnics show. Firefly flashes or bioluminescence, the molecular structure of that has actually been used for a long time in medicine. You have two molecules. You have luciferin and luciferase, and the firefly can control both those molecules, and when they come together, it creates a flash. So we've actually been all over the world collecting fireflies, and we bring them all back into our lab, and we do research on their eyes and their photic organs, which is the flashing part of their tail. And we'll sequence those genes, and we'll put those genes into a larger database of other firefly species. I've never seen that before, and I've been collecting in Utah for 10 years. My colleagues at work have been collecting for 30 plus, and they've never seen it till last year.
Okay. So as we kind of get to the end here, hopefully you recognize that there's constant new discoveries and developments in science, especially when it comes to different creatures, how they do things, and molecular biology is a powerful new tool. Tie this together with what we learned about GFP at the beginning and the fact that all species, plants, animals, fungus, are coded using the same DNA bases. We can take genes from one species and insert them into another. And we'll talk more about this when we get to genetic engineering later on this term. But it just shows you just how how closely related we are, not only to each other, but also to all the rest of life here on our planet. And so I think when we really start studying DNA and genetics and molecular biology and biochemistry, the shared nature of that is really what we need to focus on, how much we have in common, not how much difference there is, because it's a pretty tiny amount of difference when you start looking at the grand scheme of things. All right, guys, that's all I got for you today. Have a great one. I'll talk to you soon. Take care. Bye-bye.